The basal ganglia, along with the thalamus and motor cortex, are responsible for the planning, control, and execution of movement. The basal ganglia receive inputs from all cortical areas involved in motor function and channel this information back to the motor cortex through the thalamus. The main input structure of the basal ganglia is the striatum. Cortical inputs to the striatum trigger activity in the direct and indirect striatal pathways. These two pathways project from the striatum to other basal ganglia structures and achieve opposite effects on motor behavior. Basal ganglia structures are predominantly inhibitory. The striatum, globus pallidus internus, globus pallidus externus, and substantia nigra pars reticulata all inhibit their target structures when active. An exception is the subthalamic nucleus, whose activity excites its target structures. The direct pathway projects from the striatum to the globus pallidus internus and substantia nigra pars reticulata. Activity in the direct pathway inhibits the globus pallidus internus and substantia nigra pars reticulata, resulting in disinhibition of their target structure, the thalamus. The thalamus is responsible for the excitation of the motor cortex. Therefore, disinhibition of the thalamus facilitates motor cortex activity, promoting movement. The indirect pathway projects from the striatum to the globus pallidus externus, which in turn projects to the subthalamic nucleus. The subthalamic nucleus projects to the globus pallidus internus and substantia nigra pars reticulata. Activity in the indirect pathway inhibits the globus pallidus externus, resulting in the disinhibition of the subthalamic nucleus and the consequent excitation of the globus pallidus internus and substantia nigra pars reticulata. Excitation of the globus pallidus internus and substantia nigra pars reticulata inhibits thalamic activation of the motor cortex, thereby suppressing movement. A cardinal feature of the striatum is its dense dopamine innervation, which arises from the substantia nigra pars compacta. This dopamine signaling is critical to normal functioning of the striatum and basal ganglia. The two neuronal subtypes comprising the direct and indirect pathways respond to dopamine in contrasting ways due to their differential expression of dopamine receptors. Direct pathway neurons express D1 receptors, which upon activation by dopamine increase their intrinsic excitability. Indirect pathway neurons express D2 receptors that decrease their intrinsic excitability in response to dopamine. By virtue of its dichotomous effect on D1 and D2 receptors, dopamine excites the direct pathway and inhibits the indirect pathway, thereby playing a regulatory role in motor behavior. In Huntington's disease, differential vulnerability of neuronal subpopulations within the striatum results in a more profound degeneration of the subtypes that comprise the indirect pathway. The consequent disruption in striatal pathway signaling leads to pathological excitation of the motor cortex, which manifests as chorea.